And now it is my pleasure to introduce tonight's presidential lecturer, Dr. Earl K. Miller. Dr. Miller is a Pickauer Professor of Neuroscience at the Pickauer Institute for Learning and Memory and Department of Brain and Cognitive Sciences at MIT. His research focuses on investigating neural mechanisms involved in cognition, most recently observing the effects of propofol on these mechanisms and consciousness. Dr. Miller received his master's and PhD in psychology and neuroscience from Princeton University. He's received numerous prestigious honors and awards, with his most recent being awarded this year by MIT for excellence in graduate mentoring. In 2017, a paper he co-authored with Jonathan Cohen titled An Integrative Theory of Prefrontal Cortex Function was recognized as the 5th most cited paper in neuroscience. Dr. Miller's lecture today is titled Cognition Emerges from Neural Dynamics. Please join me now in welcoming Dr. Earl K. Miller.
Thank you, John. Well, thank you, John, for the invitation, for that nice introduction, and thank you all for being here. So we'll just jump right in. So this is our laboratory, and what we study is we study executive brain functions. Executive brain functions, put simply, are self-control. Our brains don't just react to the environment. We organize perception, thought, and action from within, using our goals and plans. Okay, so we don't just respond to the environment, swim towards food or swim away from light. We can actually make plans and make things come about on our own. And what we do to study executive brain functions, we record electrical signals from the brain of non-human primates. And we record 2 types of signals, which I'll talk about today, and that's spiking activity from individual neurons, as well as local field potentials, or LFPs. And these are the electric field influences that surround neurons. So all this neuron spiking is taking place in this rollicking sea of electrical influences. And then what we do is we use computational analytics and modeling to make sense of these signals. So I want to start today with a question, and that is, what is needed for executive top-down control of the brain? Well, I think what is needed is self-control requires large-scale dynamic neural self-organization. Large-scale because you have to coordinate the activity of hundreds of millions of neurons in order to do something as simple as focusing attention. Anything our brain is doing that involves top-down goal-directed behavior requires coordinating the activity of countless numbers of neurons. And it's self-organization because this organization isn't driven by the environment. It's guided by an ongoing internal top-down narrative, a model of the situation, goals, and solutions. And this is acquired through experience. So our brains learn to model the world and use that to drive and coordinate our behavior. And this model only matters if the substrate can rapidly shift processing to meet changing demands. So the brain has to be flexible enough to deal with the complex dynamic world that we created and we live in. So how does the brain do this? Well, the classic view from 20th century neuroscience, i.e. what I learned in graduate school, was the brain stores and computes information by modifying network connections. And you're probably all familiar with this. I'll be at the Society for Neuroscience meeting, but also AI is modeled after this. Changing connections, not only for storing information, but also for computation. And what I learned back when I was a graduate student was that the brain is composed of specialized neurons sending electrical impulses, spikes, those are the spikes, and they act through synapses and the synapses act kind of like logic gates. So this is illustrated by this nice figure that Nikos Logothetis published a number of years ago. And what it shows is an image on our retina, and the idea was that at the first level of processing, there is edge detectors back in your primary visual cortex. Then they converge onto longer edge detectors, which then converge on corner detectors. And there's more and more converging, and eventually you get the complex shape detectors. And there's a lot of neurons in your brain, so you do enough combination, eventually you get the peace, love, and understanding. That's the way we thought about it. And I'm not saying this is wrong. This is not wrong. A lot of things in your brain actually work like this. It's just that it's incomplete. But importantly, this view is foundational. When we first started trying to figure out how the brain worked back in the 20th century, you don't start by jumping in the middle to the most complex behaviors and complex functions. You start on the edges. You started with the inputs and the outputs. And this model was based largely on the sensory motor perspective, starting to figure out the brain from the edges. But around the turn of the century, several of us, my lab included, several others, began to study things beyond just simple sensory motor functions, just connecting a stimulus to a perceptor, a stimulus to an output. We started training animals to perform behavioral tasks based on acquired knowledge, categories, rules, or principles. So for example, we taught monkeys to categorize a bunch of different images as cats versus dogs, and the animal had to ignore what the individual cat and dogs look like and just tell us what group that individual belonged to. We also did things like train animals to learn new categories based on category prototypes. We taught animals to make number judgments, small numbers 1 through 5. And we taught animals things like rules and principles, like same versus different, or match versus non-match, or up versus down. And all of these tasks had the same thing in common. They all emphasized top-down information over sensory motor details. The animal didn't need to tell us what the individual cat or dog was, or what the individual display showing a number is. They would tell us that stimulus was a cat or was a dog, and what number that was. So all these tasks emphasize that top-down information at the expense of sensory motor details. And they were recorded in the prefrontal cortex, the top of this hierarchy where all this combining stuff is supposed to go on. And what we found is that neurons in the prefrontal cortex acted like filters for top-down information. Their spiking reflected categories, rules, and other task-relevant factors, and showed little sensitivity to irrelevant sensory details. So neurons in the prefrontal cortex would tell us whether a stimulus was a cat or dog, and wouldn't convey any information about which cat or dog it was, but the individual. And also, this top-down information was distributed across many neurons, not a small number of specialized neurons, but as many as a third of the population or more. So, first question is, what is the prefrontal cortex doing with all this top-down information? Well, in 2001, Jonathan Cohen and I proposed the integrative theory of prefrontal cortex function, where we proposed that the prefrontal cortex builds models of successful goal-directed behaviors, and then it activates those models and maintains these activity patterns that represent goals and the means to achieve them, and that sends feedback signals that flow back to the rest of the cortex and bias the flow of activity so the cortex can produce the appropriate behavior, appropriate goal-directed behavior. But all this work suggested that cortical processing is not really a drive towards small populations of highly specialized neurons. We did not see that when we looked at the top of the cortex. Instead, it seemed to be an experience-dependent dimensionality reduction, a distillation of information down to its goal-relevant essence. And Lisa Feldman and Baird and I just wrote a paper describing that we think this is the main function of the brain, is to do dimensionality reduction to the essence of the information needed. But importantly, we also found this information was distributed across many neurons that had a property that we now call mixed selectivity. These are neurons that seem to be multifunctional. They were not specialized. And they were as many as a third to half of randomly selected neurons in the prefrontal cortex and other higher cortical areas. So let me show you what this property of mixed selectivity is. Here's a simple task. Remember 2 pictures and their order. The monkey sees 2 pictures in this case. In this case, it's what's shown as a butterfly, then a padlock, and then at the end of the trial, 3 stimuli appear, and the monkey has to saccade to the 2 pictures that he saw just before in the correct order. So you train animals on a task like that, and the classic pure selectivity version of the way we used to think about the brain would predict this kind of a pure selectivity where neurons respond to independent factors or just a simple combination of factors. So shown here in the bottom is an example of a neuron, and here's the neuron responding to stimuli, these objects, when they're presented first versus second. Again, the monkey has to remember the order of the stimuli. And what the classic model predicts is that neurons that are selected for an object will always respond to that object in the same way. So this neuron I've illustrated here is responding most to the padlock and least to the butterfly, and it does that whether the stimuli with the objects are presented first or second, doesn't matter. And there might be another population of neurons that just convey the order. Some neurons that will respond more when a first stimulus appears, and then other neurons will respond more when a second stimulus appears. And somehow these two factors combine, and you get performance of the task. So that's a very simple model based on the classic view of the brain. But when you actually record in the prefrontal cortex, what you find is something very different. You find this property of mixed selectivity. These are neurons that change their selectivity with context. So in this case, now this neuron, this mixed selectivity neuron, is responding most to the padlock when it appears first and least to the butterfly when it appears first. Then that selectivity reverses with the object appears second. It responds most to the butterfly when it appears second and least to the padlock when it appears second. So what these neurons do is they're spiking the nonlinear combinations of task factors. And this gives them context-dependent selectivity. This neuron essentially is showing different selectivity, conveying different messages in different contexts, first versus second. And because they're context-dependent selectivity, this makes them multifunctional, or doing different things in different contexts. Now, why are there so many mixed selectivity neurons in higher cortical areas? Well, my colleagues Maria Rigatti and Stefano Fusi, using data that Melissa Warden collected in my lab, they built a computational model that answered this question. And what they showed is that mixed selectivity is critical for cognition because they provide a nonlinear, high-dimensional representational space that gives the brain extra computational horsepower. By having this nonlinear mixed selectivity, the representational space is complex and it conveys a lot more information. And also, this increases the brain's storage capacity because now instead of the information being stored in just a few specialized neurons, it's distributed across many neurons, which essentially renders the system capacity unlimited, virtually capacity unlimited. And again, because they're doing different things in different contexts, this is a substrate for flexibility, and flexibility is what cognition is all about. We don't respond the same way in every situation. We respond differently in different situations. If there was one word, if I could substitute one word for cognition, it would be flexibility. And that's what these neurons seem to do, seem to be that substrate. But now let's go back to the question I asked at the beginning of the talk. What is needed for this top-down executive control, the self-control of the brain can control itself? And I propose that it requires large-scale dynamic neural self-organization. Well, note that mixed activity means that cortex is not made up of fixed, specialized neurons in rigid networks. We originally found these neurons, these mixed activity neurons, in the prefrontal cortex, but since then they've been reported all over cortex and also subcortical structures. They seem to be rather common in the brain. And this suggests that cortex is not made up of these fixed networks doing specialized things. Instead, it consists of many multifunctional neurons sharing overlapping connections, network connections. And this means that neurons dynamically shift their network membership depending on context. So neurons are not part of just one ensemble or one network. They participate in many different ensembles, and they shift their ensemble or network membership depending on the context. Now think about how difficult it would be to organize a system like this at the level of synaptic connectivity alone. For you younger people in the audience, this is how telephone calls used to be made. Someone had to actually physically move a connection from one socket to another in order to connect your call. But think about if you had to organize the brain, this large-scale dynamic neural self-organization, organize the activity of a gazillion neurons. How do you do that in the synaptic level? You have to somehow control and coordinate a gazillion synapses on a gazillion neurons. It just seems computationally intractable. And also, spiking in synaptic transmission is relatively slow. It takes about 100 milliseconds for a sensory input to activate neurons in your frontal cortex. 100 milliseconds isn't a long time if you're just simply responding to the environment in a reflexive way. But it's a long time constant if you're doing real computation. So what's the solution? How can the activity of millions and millions of neurons be organized on the fly in this way? Well, it may be the next level up of processing. And that is, brains produce oscillating electric fields, we're calling brainwaves. Brainwaves are an emergent property. You see on the bottom there's a crowd doing the wave. You see the wave moving from right to left across the screen. Now these sports fans in a stadium are organizing themselves just based on a few simple rules. And if if they can organize themselves that way, in this emergent way, imagine what evolution could do with that principle. And in fact, brainwaves are organization. Networks of recurrent connections will naturally begin to oscillate, and that's an internal organization. I can imagine, very early on, very rudimentary nervous systems, simple creatures are just responding towards food or swimming away from light. When the nervous system evolved to get complex enough to begin oscillating, well, as soon as the neurons are oscillating, that's an internal organization. Now the network is no longer, the nervous system is no longer just responding to the outside world. It has its own internal organization. And it may be a dumb organization, just simple oscillations, but evolution could have evolved mechanisms that push around these oscillations, take advantage of this natural internal organization and learn to push around the oscillations and produce control. And let's not forget that these oscillating brain waves are cycles and patterns of excitation and inhibition. That's what an oscillating wave electric field is. It's excitation and inhibition, and that's how the brain works. But furthermore, wave dynamics makes this large-scale dynamic self-control, it makes it much easier. Because waves are computationally tractable. A simple wave function is a very simple function, and it tells you the wave's action over time and over distance. And furthermore, these waves operate on the mesoscale level, on the order of a spatial scale of hundreds of millions of neurons, which is just the scale you need for top-down control. And electric field influences are continuous. They're happening all the time. I'll get to this in a moment, but cortical neurons don't really fire that often. And plus, neurons are fickle, noisy, and unreliable. You get something called representational drift. If you record from neurons in cortex and present the same stimulus 20 times in a row, you'll get 20 different bursts of activity from these neurons. They'll give a little burst of activity, then they'll be quiet for a while, then maybe next trial they won't fire at all, then maybe they'll give another little burst of activity. It's like the cortex is the world's largest orchestra, and the song is playing on, but individual players, individual neurons, are playing a little snippet of the melody, then they're quiet for a while, then they play another snippet of the melody, and they're quiet for a while. They're only contributing every once in a while. So if you want to organize the brain, you don't want to act on the level of these fickle individual players, you want to act on the level of the sections, you want to act on the group level. And plus, electric fields provide fast influence. Electric fields spread influences around the brain 5,000 times faster in synaptic transmission. And that sounds like a pretty good property for large-scale organization of the brain. So I'm going to argue that large-scale dynamic neural self-organization must happen at this level of emergent level of brainwaves. Now, Dimitris Panosas showed that you can actually read information from the brain using the electric fields alone. This is a study where the non-human primate was held 4 different objects in working memory. And what Dimitris showed is by recording from the LFPs and the near electric fields just hovering just above the electric fields, you can actually read the contents of working memory, what stimulus, what object the animal is holding in mind in real time. And with no representational drift whatsoever, just a pure, steady informational signal. So that sounds like a pretty good way of studying the brain. You have this pure informational signal, you can study the level of the electric fields. But my attitude is that if there's something that's useful for us to figure out the brain, evolution probably took advantage of it. And it can because there's something called Ephaptic coupling. Electric fields have a causative influence in the brain. We showed this, Demetrius and I showed this in our paper, but this is many, many others. If you go do a Google Scholar search on Ephaptic coupling, you'll find thousands of papers demonstrating electrical influences inside the brain. And the way it works is that spiking generates fluctuations surrounding electric fields. And then they turn, those electric field fluctuations turn around, they influence spiking. That creates more spiking, which causes more electric field influence, and so on and so forth. This iterative process. Spikes influencing electric fields turning around influencing spiking. And this works because cortical neurons don't spike that often. Cortical neurons give a little burst of spiking, and most of the time they're silent. Cortical neurons fire way below their capacity. The average cortical neuron fires about 15 hertz. And again, only gives periodic bursts of firing. So cortical neurons are really only spiking maybe like 20% of the time. 80% of the time they're silent, not spiking. So they spend much of their time with their membrane potentials oscillating, teetering into the spiking threshold. Remember, their membrane potentials, these oscillating electric fields, are driving the membrane potentials up and down, up and down, up and down. So as a result, neurons are mostly spend most of their time in the state where they're teetering on the edge of spiking. Am I going to spike? Am I not? As these electric fields are oscillating. And all it takes is a small change in that electric environment to either push them to spike or push them not to spike. And in contrast, electric field influences are continuous. They're happening all the time, not 20% of the time, but all the time. And also, astrocytes, I'm learning more and more about how astrocytes having a direct role in function and behavior. Astrocytes are about half of the cells in your cortex, and they have electrical properties like neurons. They have membrane potentials. They are affected by electric field influences, and they spread electrical field influences. But what astrocytes don't do is they don't spike. So what that means is that a lot of cortical activities on the electric field level. Neurons are half the cells, and they're spiking 20% of the time, but 100% of the cells in your brain are oscillating in this electric field level 100% of the time. So a lot of cortical activity is on the electric field level. But I want to make the point here, it's not a dichotomy. It's not spikes versus electric fields. They're all part of one integrative system. There are different ways of measuring the same system. Spikes create the electric field changes. Electric field changes turn around and influence the spiking, and so on in an iterative process. So electric fields, because of their very nature, they exert mesoscale level influence, the scale needed for top-down executive control, the level of hundreds of millions of neurons. But electric fields can also have an influence on the microscale. Dimitris Panosas and I, and Gene Freeman and I proposed the cytoelectric coupling hypothesis. And what we suggest is that electric fields can restructure the components of the cytoskeleton, these are proteins, filaments, microtubules, like grains of sand on a speaker organized by sound. You see on the bottom here, you probably have all seen this, you pour a bunch of sand onto a speaker and you play a note, and the sand organizes itself into a pattern. So we think that electric fields can do this on the micro level in the brain, restructuring the cytoskeleton of the brain on that level. And what this does is this tunes networks to oscillate more efficiently. Biology loves energy efficiency. So this oscillating activity allows the networks to oscillate in a more robust way using less energy. But because oscillations carry information, as I just mentioned, this means that it also tunes networks to process information more efficiently. So granted, this is a molecules to mind or mind to molecules framework. These same oscillations that can play a role controlling the brain and carrying information can also tune the networks to process information more efficiently. It's a way the information the brain is processing to tune the brain on the fine scale. And you think that sounds a little wacky? Let's remind us that developmental biology tells us that electric patterns within tissues act as a blueprint during development. How do the cells in your hand know when you're developing? How do they know to make the shape of a hand? What they do is the body sets up electric field gradients in the shape of a hand, and the neurons migrate along the shape of the hand and form your hand. So that's happening on the structural level in development. Why shouldn't it happen inside the brain, which is an electrical system to begin with? So over the past 10 years or so, our lab has amassed right now a considerable amount of data suggesting that there's a major division between the higher frequencies, gamma and above, and the lower frequencies, namely alpha, beta, about the mid-level frequencies. And what they do is they have two general types of different functions in the brain. We've shown that that gamma, and coupled with spiking, is feeding forward sensory information from your sensory cortex forwarded to your brain to your frontal cortex. At the same time, flowing in the opposite direction are alpha-beta influences, and they seem to be inhibitory to the gamma. They inhibit gamma, suppress gamma, suppress spiking. So these alpha-beta control signals, feedback signals, may be a way for top-down signals to control bottom-up, to filter bottom-up information as it's flowing into your cortex, thereby regulating sensory processing and preventing sensory overload. We've shown that in a series of papers that the top-down alpha beta is primarily is strongest in deep layers of the cortex. The deep layers of your cortex are the feedback layers of cortex. So if they are playing this top-down control, if alpha beta is playing this top-down control role, they should be stronger in the deep feedback layers of cortex. And we show that these top-down alpha beta, the deep cortical layers, then suppresses bottom-up gamma spiking in superficial cortical layers. Now we devised this model from recordings in the prefrontal cortex. But since then, Diego Mendoza-Holiday and Alex Major, Diego from Bob Desmont's lab, Alex Major from my lab, showed that the spectrolaminar motif with more gamma, stronger gamma in superficial layers of cortex than stronger alpha beta in deep layers of cortex, this spectrolaminar motif is ubiquitous in cortex. We found it in 20 plus cortical areas in 4 different species. And it's so robust, you could find the middle layer of cortex just by seeing where the alpha beta gamma crossover is. And when something is that robust and ubiquitous throughout cortex, it must be doing something important. So this idea of bottom-up content from the outside world being carried by high-frequency gamma and it being excitatory, and it's matched and regulated by this top-down alpha beta, which is inhibitory, was recently incorporated into a model which we call spatial computing. Michael Lundquist created this model. And what it suggests is that the brain is using negative network space, non-activity, inhibition as a coding dimension. And the idea is captured here. So what the idea is that these alpha beta influences are patterned. They're not just a blast of alpha beta, they're patterns. And they essentially form a top-down inhibitory stencil, or a photographic negative, if you want to think about it that way. And that tells which parts of the cortex are not allowed to activate. And then when gamma, carrying information from the outside world, it sort of fills in the gaps there in the stencil, creating a pattern of activity that's defined by the alpha beta stencil itself. And then by controlling which patches of cortex are allowed to become activated, that's how the brain controls its own processing. And I consider this to be an update to the Miller and Cohn model of 2001. It's the same basic idea. The prefrontal cortex is providing this control that guides activity of the rest of the cortex. But back in 2001, we thought about it in terms of spiking and synaptic connections. But now we think that the control is coming through these inhibitory alpha beta patterns that create this top-down stencil in cortex. Now, this concept of spatial computing can also explain mixed selectivity. So here's our example of mixed selectivity neurons again. We have these neurons that show this nonlinear context-dependent spiking. Neurons change their selectivity from, in this particular experiment, on context. They have a different selectivity when something is presented first versus second, so the context matters. And the idea here is that these two contexts, these two top-down contexts, first versus second, which is the requirement of the task, remember their order, are conveyed by 2 different alpha beta patterns for the context. There'll be one alpha beta pattern for the context of being first, and another alpha beta pattern for the context of being second. So in other words, different stencils for the context of being first versus second. And as a result, excuse me, different neurons will respond in different ways in these different contexts according to the different stencils. So alpha beta patterns control which neurons can spike in which context. That's the general idea. And as a result, you get neurons with nonlinear context-dependent spiking. They will respond like different neurons in different contexts because of this alpha beta inhibitory influence. And coming along with that are all the computational benefits of mixed selectivity. Now, in another line of work we're conducting with Emory Brown's lab at MIT, we're studying how general anesthesia causes unconsciousness. And there was a tacit assumption for a long time that anesthesia just simply shuts off your cortex. But that's not what happens at all. It drastically alters these brainwave dynamics. So what general anesthesia does is shift cortex to low frequencies. This is an example of on the top of the panel there on the left are a bunch of LFP recordings from 4 different cortical areas. And the bottom are spike rasters showing that the spikes line up with the LFPs. And what you can see is that this is now after unconsciousness. In the awake brain, the frequencies they're high frequency and they're low amplitude because all these neurons are doing different things and they're activating different ways. But once general anesthesia makes you unconscious, the whole brain shifts to these low frequency delta waves. Okay, and not only that, but they also scramble the phase alignment of these waves. So we recently discovered last year that it isn't just a shift to these low frequencies. The low frequency waves, then everything below 30 hertz, then shifts, their phase alignment flips almost 180 from the awake state. So anesthesia isn't just shifting your brain to low frequencies like sleep, it's also scrambling the phase alignment of the brain waves. And if the waves are misaligned, that's going to break up cortical communication. It'll prevent cortical communication and render unconsciousness. And the third thing it has, which is related to the first two, is it all destabilizes cortex, makes the cortex more wobbly and unable to keep on the correct path of computation. And the reason why I mention this is because we found all three of these effects. In 3 different anesthetics, they have 3 different molecular actions. Propofol, which is a GABA agonist, ketamine, which is NMDA, and dexmedetomidine, which is alpha-2. These 3 different anesthetics act on different receptors in the brain, on different neurons in the brain. Propofol is inhibitory, the other 2 are excitatory, mixed excitatory, so they're doing different things to neurons in the brain. And if you ask some of my more reductionistic colleagues, how are these 3 anesthetics making you unconscious? They would have to say, oh, it must be 3 different pathways, 3 different effects, because they're 3 different molecular actions. But it turns out that all 3 of these drugs are doing the exact same thing. They're having all 3 of these effects, and they're doing so identically. If you even look at the data, you can't tell which drug the animal is under just by the data alone. So what this says to me is that there's an emergent level of wave dynamics can explain something that reductionist approaches can't. We can explain how all these anesthetics all do the same thing to you, and something that can't be explained on the more detailed level. And to me, that's a pretty compelling evidence that this emergent level of wave dynamics is doing something important, having a real important function in the brain. Now, these waves don't just stand in place like a jump rope. They're not standing waves. They're constantly traveling and moving around your cortex. A recent example of this is just published last month from Hyobin Han in my lab. Hyobin was studying monkeys performing a working memory task that involved, it's called a change detection task. What happened was the animals where the red circle is where the animal's eye is, and the animal fixated a spot of light. Then an array of 2, 3, 4, 5 colored squares come up, goes away. About 800,000 milliseconds later, the array comes back on, but one of the squares has changed colors. And the monkey has to tell you which square changed colors. Well, what Hyobin found was that target detection, the ability to detect that square that changed colors, varied with the phase of theta waves. Now, we're talking about lower frequency waves now, 3 to 6 hertz. The target detection varied with the phase of theta waves in frontal cortex. During this memory delay, there's ongoing theta waves that's intrinsic to the cortex. And then the test array, the second array where the animal's got to make the judgment and find the target, is happening at some random time relative to that ongoing theta wave. And depending on when the test array appears, when the animal has to judge, you get either an increased accuracy or decreased reaction time, depending on the phase of theta wave. So what's shown here on this plot here is green is reaction time. The black line is accuracy, how well they performed. For reaction time, up is bad, down is good, slow versus fast, and for accuracy, the opposite. And what you see on 0 there is the phase of theta when that second test array appeared, what phase theta was at. And you can see there's a clear change in reaction time and accuracy depending on where that theta wave was. But moreover, Hyobin found that target detection also varied with where the target appeared relative to the theta wave from the top to the bottom of the display. So on the top plot here, you see that if the target was near the top of the display, then if the theta wave if the test array appeared when the theta wave was at its peak, then you get increased accuracy and decreased reaction time, better performance. But then as you move down that row here, what you see is that depending on where the target was, the theta wave coming progressively later and later and later and later is better for targets that are lower and lower and lower in the screen. So to put it simply, what seems to be happening is it seemed like there was a theta wave. These are recordings from the frontal eye fields, I should mention. And what seemed to be happening is there seemed to be a theta wave sweeping across the cortex, as if a theta wave was scanning the mental image of this array that the animal is holding in mind, constantly scanning it 3 to 6 times a second, like radar. And then how the target and the theta wave lined up determined how the animal's behavioral performance. Now, you mentioned this is theta wave. This is lower than the alpha beta I told you about earlier, which is around 15 to 25 hertz. But theta seems to be almost like a master clock that helps control what the other waves are doing. And when we looked at this theta traveling wave, we found these cross-frequency interactions. The excitatory versus inhibitory phases of theta showed increased gamma versus alpha beta. Increased gamma during the excitatory phase of the theta wave, traveling wave, and increased alpha beta power, the inhibitory alpha beta power, during the inhibitory part of the theta traveling wave. So it's as if this theta wave was dragging this alpha beta dynamic, this alpha beta push-pull dynamic across the surface of cortex. Now, another way these traveling waves can do, what else these traveling waves can do is they can organize and maybe even perform computation. So here's an example. Now we're back to neuron spiking now, and this is an example of subspace coding. Subspace coding is, if I record from 1,000 neurons simultaneously, I can create a 1,000-dimensional space that describes what those 1,000 neurons are doing. That's self-evident, it's self-descriptive. Then you ask the question, how many dimensions does it actually take to explain what these 1,000 neurons are doing? And the answer is you can do it in just 3 dimensions. Now, this is a recording from a couple hundred neurons during a working memory task. And we've now done this dimensionality reduction in the space of explained variance. And we could see that in the subspace, there's information about the object. So ignore the rotation for a moment, but the 4 colored lines correspond to 4 different objects the animal's holding in working memory. And you see the separation between the lines. That means there's information in this pattern of neural spiking about the object being held in working memory. What's happening at time 0 is in the middle of the working memory delay, we distract the animal by showing it a distractor, or have the animal move its eyes to create a distraction, something that disrupts the animal's performance by about 15%. So what's happening at time 0, the distraction appears, and what goes on is the neurons get knocked out of the space they were before by the distraction. Then the neural patterns gradually and smoothly rotate back around to where they were before the disruption. So this rotation in subspace, these smooth changing patterns of activity are showing the recovery. The rotation is bringing the neuron patterns back to where they were before the disruption. And sure enough, if the rotation isn't complete, the animal showed errors. If the rotation isn't complete, the animal is more likely to perform the task correctly. So subspace coding means that if you could record from hundreds of cortical neurons and you could explain them by only 3 dimensions, that means cortical spiking is highly coordinated, not like a bunch of insects flittering in the night sky in some chaotic way. Instead, cortical spiking is more like a murmuration of birds flocking together in the sky, all moving together. At least in terms of the shared variance space, that means cortical spiking is highly, highly, highly coordinated. Okay? And the question is, how does the brain produce this kind of flexible organization? The question I asked at the top of my talk. Well, when I see neurons behaving in a highly coordinated fashion, like birds flocking in the sky together, the image that comes to my mind is a wave moving across a network of neurons. And sure enough, that's what Tamal found. He found when you look at the electric fields in the same cortical areas, the same recordings, where we found this rotating subspace dynamic, we found rotating traveling waves across the surface of cortex. So what's shown here is recordings from an array of electrodes in the prefrontal cortex. Each square is on a different electrode. The color is voltage value. And you can see it in correspondence with this rotations in subspace, in this mathematical representation of spiking patterns, we see there's a corresponding rotation in electric field traveling waves, rotating traveling waves across the surface of cortex. Now let me emphasize that. What we're finding is a mapping of a mathematical construction of neural computation. This movement of neurons through subspace, these shifting patterns of activity are the neurons doing computation. You see the same path being taken through subspace for the same type of trial consistently. So it represents the neuron, it's reflecting something about neural computation itself. But there's really no reason in principle why this rotating movement in this mathematical representation of subspace should correspond to an actual rotation across the surface of cortex. What this suggests to me, the possibility that there's a role for these waves in not just organizing neural activity, but performing computation, something called analog computation. So what's analog computation? Here's an example of analog computation. You see two waves moving in opposite directions and colliding together, the blue wave and the yellow wave. And the dotted line shows the summation of the two waves as they travel past one another. And where the vertical dotted line is where you can sum anywhere along space, and you can see that you get simple addition and subtraction. These two waves moving past one another are doing addition and subtraction. And this is just one dimension and one frequency, just two identical sine waves, one frequency, and just moving together in one dimension. Your brain, because all these traveling waves are happening across multiple frequencies, and they're in 3 dimensions, your brain can do this kind of computation in 3 dimensions and across multiple frequencies simultaneously. That's a lot of space for computation, a lot of opportunity for computation there. And this idea of analog computation is not hypothetical. It's not maybe. It's not only it's been used for a very long time. For example, there's a tide predicting machine that predicted tides by using wheels and knobs back in 1872. Vannevar Bush from MIT built an analog differential analyzer in 1927 to calculate missile trajectories. And it used wheels and disks to solve 18 variable differential equations. That's some pretty complex math. He did it all with just wheels and disks. But wheels and disks and oscillating electric fields are the same thing in principle, only electric fields are more easy to manipulate. And one of my favorite examples is back in the 1980s, George Gerstein built an all-analog principal components analysis for sorting complex waveforms down to their principal sine waves. And he did it using a bunch of wires and capacitors and resistors and a waveform generator to do analog computation. And that principal components analysis was that his analog device could do was way faster and less power hungry than the state-of-the-art PDP-11 digital computer that was state-of-the-art at the time. And I mention this because I use this for my dissertation work. It's near and dear to my heart. But this isn't just from the past. This is happening now. People have shown that you could build recurrent neural networks, then let them produce the waves, and then by doing a few easy manipulations in the network, you can actually have the waves come together in a way that can do real computation. And you can also do this with light waves too. It's the same thing. As long as the wave has an oscillating component, you can do analog computation with it. Now why? So the brain has these waves, analog computation is possible, but why would evolution want to do analog computation? Why would it have lighted on that particular solution? Well, why analog computation? Analog computation is information rich. It uses continuous signals. The real world is continuous signals. Digital computing breaks the world into little ones and zeros, which is not like the real world at all. And if you think about these waves as the peak of the wave being 1 and the trough of the wave being 0, you have all the values in between the 1 and the 0. So it's information rich. And that makes it very efficient. But what makes it super efficient is that analog computing is inherently parallel. So the way Vannevar Bush set up is he had to model different differential equations. You set how the wheels and disks, how they relate to one another, how you set them in different opposition to one another. You set them up in a different way, and that models the equation. The actual geometry of how the wheels and disks fit together models the equation. And then if you want to solve the equation, you turn the wheels, and the entire equation gets solved in parallel because all the variables, how the waves come together, how the wheels and disks come together model the equation because all the variables are there in the waves and in the wheels and disk. As soon as you turn the wheel, then the entire equation gets solved in parallel. Whereas digital computing is inherently sequential. That makes analog computing way more energy efficient. Analog systems are vastly more computation and energy efficient than digital. And when I say vastly, I could say vastly, and still leave a few vastlies to go. Because AI consumes enormous amounts of energy. They're building nuclear power plants, hydroelectric plants to power AI. They're putting these data centers in these rural areas, and they're causing blackouts and brownouts in the surrounding towns, and also causing droughts because it takes a lot of water to cool these computers that are doing digital computation. So AI consumes enormous amounts of energy, and it ain't that smart, right? So biology, evolution favors energy efficiency. Biology does not solve problems by throwing tons of energy at it. In fact, a pretty good definition of life is local reverses of entropy using as little energy as possible. And when you consider that, consider the fact that the human brain, which is far more capable than the smartest AI, runs at about the power it takes to light a dim light bulb. So clearly, I think, brains are doing something fundamentally different from the way from digital computation, doing something fundamentally different from the way AI approaches the problem of intelligence and computation. Okay? And your brain is constantly generating the raw materials for analog computation. These organized traveling waves are exactly what you need for analog computation. So if evolution didn't take advantage of this, then I don't understand evolution. So in conclusion, I think cortex is a hybrid system. Connections can store information, and probably relatively simple forms of computation can be done by connections. But I'm suggesting that the real high-powered computation is done by waves. And the idea is that information stored in the synaptic connections, that's how the brain stores information, produce spiking patterns. Different information will produce different spiking patterns, and that creates different wave patterns. Then those wave patterns turn around and influence the spiking, and so on, and it generates on and on. The spiking creates the rollicking of the sea, then the sea sort of modulates the spiking, and then creates more spiking, which then rollicks the sea a little more. But importantly, waves are a computationally tractable way for a brain to achieve executive control over their own processing. Waves are very simple functions. They operate just on the scale you need to produce real control on this mesoscale. But also, they're a computationally tractable way to compute. Analog computing is information-rich and parallel, and thus highly energy efficient. And biology loves energy efficiency. The final thought I'm going to leave you with is that consciousness may be a natural outcome of analog computation. Most biological theories of consciousness propose that consciousness is a result of a meta-ensemble, a large network that somehow unifies cortex, creating a unified experience. Now we know this because consciousness is a unified experience. Something's got to be unifying all that information into a unified whole. But also, consciousness is good for flexibility and planning, and that kind of big picture stuff needs a unified representation. So what I want to suggest is that consciousness arises when these analog computations create wave patterns that eventually become large. These analog computations can happen on both a local level and then feed up to a higher level. And when the analog computations create wave patterns that are large and organ enough to unify cortex, you get consciousness. Okay? So maybe that's what brain processing is. It doesn't matter, you don't need a homunculus to interpret what's going on in the brain. You don't need a downstream decoder to figure out what spikes are doing. You simply have wave patterns begetting new wave. As the waves come together, they create new wave patterns in computationally principled ways. And those wave patterns are patterns of excitation and inhibition. So excitation and inhibition, patterns coming together and creating new patterns in a computation principle way using analog computation, be all there is to brain processing, waves and patterns begetting waves and patterns. No reason for anything else. So I thank you for your attention, and I especially thank my laboratory who do all the hard work on this.
So thank you so much, Earl, for an amazing talk. We can now take